All right, we have talked about how you control when proteins are made and when they're not made in prokaryote cells, which is very, very simple compared to eukaryotic cells because prokaryotes are simple. They're one cell, but eukaryotes generally, very often, are multicellular. So you've got to consider not only how one cell can shut certain things on and off, but how all of these cells can communicate with each other and interact with each other, and that my skin cells have a bunch of genes in them that must be turned off because I don't want my skin cells to become nerve cells like in my brain, and I certainly don't want my nerve cells in my brain to become skin cells. So some of those genes have to be permanently silenced, while some genes we only want temporarily silenced under certain conditions. So there's a whole level of complexity to eukaryotic gene control that is not present in prokaryotes. Okay? So, there are many points of control in eukaryotes where we can control gene expression. Some of them are at the DNA level. Some of them are at the level of transcription, RNA processing, moving the RNA or not out to the cytoplasm, translation, the protein itself can be controlled. So there are so many points throughout, if you look at this, where we can, where we can seek to shut down or activate the expression of genes. So many places where we can control, and that's a good thing for a complex creature like you. DNA is packed. We know it's coiled and folded and so on and fit into the nucleus. And depending on whether or not we allow it to be unpacked, we can control whether it gets activated and transcribed or not. Okay? So we have a lot of information here, but basically the bottom line is if we tightly wrap this so that the RNA polymerase can't act on it, there's not going to be any transcription and the genes will be turned off. Okay, whereas if we allow it to be more loosely packed, it can be transcribed and the genes can be turned on. Okay, also, if we add certain chemicals to the DNA, we can block the transcription factors that would allow RNA polymerase to bind. Methylation, adding a methyl group. Okay, there's a methyl group right there, it's CH3, a really simple little chemical group. But if we add that to a DNA nucleotide, it's not going to allow transcription. It will block transcription factors from binding and just the genes are turned off. And that tends to be a pretty permanent inactivation of genes. So that would shut down my skin genes and not allow them to ever become active in my brain, for example. Okay. Opposite side of that, if we add a different type of chemical group to the DNA, we can encourage it to unwind and be more loose so that the RNA polymerase and transcription factors can get to it. So acetylation of the histones, those spools that the DNA rides on, can help to turn genes on, help to make them accessible to RNA polymerase and so on. We can control whether DNA gets transcribed by that promoter region. We can block it or unblock it, kind of similar to what we've talked about with the prokaryotic cells and their promoter region. We can enhance binding or inhibit binding of RNA polymerase and so on, and I'm not going to go into this detail. We can control it after transcription. We can alter how things are edited and so on and create different kinds of proteins. We can allow RNA to be degraded more quickly or less quickly. So mRNA can last just a few hours, or it can last weeks, and how long it lasts can determine how active that genetic information is in the cell. There's a really hot area of research called small interfering RNAs, or siRNA for short. There are these little pieces of RNA that can actually act like a death tag to the mRNA molecule. They'll bind to it and cause it to be double-stranded, and if it's double-stranded, then it can't be read, and that can just silence that RNA without dealing with the DNA at all. It just makes the RNA not work. We won't go into all that detail. We can block translation. We can make it so that we can't translate. And then if we've translated, we can make it so that the protein gets processed, either to become active or to be degraded or something, or inactivated so that it can't be used. So there's all kinds of ways to work with eukaryotic gene regulation, many more than in prokaryotic gene regulation. Okay, so that's going to be it for this podcast. Thanks for watching. I will catch you in the next one. Take care.